DNA extraction is one of the most modern practices of the biological sciences and have endless applications. Most of them we can't even imagine yet. In plants, DNA extraction is nowadays essential for the identification of diseases, fortification of crops, and conservation and identification of species, among many others. In this practice, we extracted DNA from cedron tissue following the CTAB protocol, where the grinded tissue sample from cedron were treated with different chemicals to get the final DNA. The materials used in this practice are extraction buffer composed of sorbitol, 3-sodium chloride, EDTA, twin, Triton X100, beta-mercaptoethanol, and water. This buffer contains detergents that help to permeabilize the cell membrane because of the disruption of the hydrophobic-hydrophilic interactions among molecules. Detergents are used to lyse cells, release soluble proteins, solubilize membrane proteins and lipids, among others. Chloroform isoamyl alcohol is also used to promote the partitioning of lipids and cellular debris into the organic phase, leaving isolated DNA in the aqueous phase. Also, sodium acetate is used, as well as ethanol 70%, ethanol 95%, a lysis buffer composed of 3-sodium chloride, EDTA, sodium chloride 5M, CTAB, Triton X 100, beta-mercaptoethanol, and water. Cold isopropanol is used to precipitate DNA. Mortars or pestles, plastic tubes, centrifuge, and a waterbed. Here you can see we have different micropipettes for the measurements of the different solutions according to the stipulated measurements needed. Here we also have little plastic tubes where we're going to put our solution on our tissue for the DNA extraction. And finally, here we have the tips of the micropipettes that we are going to use.
The first step was to add 20 milligrams of cedron samples in each tube.
Here you can see the cedron tissue. It's already grinded and it's ready for our use.
600 microliters of extraction buffer were added to the sample tubes using a micropipette. Both tubes were mixed with a vortex 3 times for 8 seconds each. Then, samples were weighted in order to have the same mass in both sample tubes before doing the centrifugation process.
Immediately after, both sample tubes were centrifuged during 10 minutes at 11,600 rpm. The samples were put inside facing each other to create the pellet in the correct side.
Next, the supernatant was removed from both sample tubes. 400 microliters of lysis buffer were added. And then each sample was mixed by inversion 8 times. Then they were submitted into the vortex 2 times for 8 seconds each.
After this, the sample tubes were placed inside a water bath at 65 degrees Celsius for 30 minutes. Every 10 minutes, the samples were mixed by inversion. At the end of the laboratory session, the samples were left in the freezer for 24 hours.
500 microliters of 24 to 1 chloroform isomylic alcohol were added to both samples and then mixed by inversion 8 times, immediately after they were put back into ice. After that, samples were weighted in order to be centrifuged.
The conditions for the centrifugation were 10 minutes and 11,600 rpm and 4 degrees Celsius. 200 microliters of the aqueous phase were removed from each sample, taking care of not removing the pellet as well. Then 200 microliters of 24-1 chloroform isomylic alcohol were added. Both samples were put into the centrifuge for 5 minutes at 8,700 rpm.
200 microliters of isopropanol and 10 microliters of 5 molar sodium chloride were added in both sample tubes.
The samples were put into an incubation process at -20 degrees Celsius for 1 week. After samples were removed from the incubation process, they were centrifuged during 5 minutes at 8700 rpm. The supernatant was removed from both tubes and they were left to get dry, leaving the pellet on the tube. After this, 100 microliters of DE buffer, 10 microliters of 3 molar sodium acetate, and 1 microliter of RNase were added to the sample tubes. The two samples were introduced to the incubator at 37 degrees Celsius during 30 minutes. Then, 330 microliters of absolute ethanol were added in both samples. Both tube samples were put into a centrifuge at 8700 rpm during 3 minutes. The supernatant was removed from the tube samples and pellet was left on the tubes. After this, 100 microliters of DE buffer, 10 microliters of 3 molar sodium acetate, and 330 microliters of ethanol 70% were added into the tube samples. The sample tubes were centrifuged at 8700 rpm during 3 minutes. At the end of this lab session, the supernatant was removed and then put back to the fridge for 5 days. 330 microliters of ethanol 70% were added to both samples after 5 days of freezing. This was made in order to wash the samples. The samples were centrifuged during 3 minutes at 8700 rpm. The liquid was piped off from the tube samples. The tube samples were placed into the water bath for 30 minutes in order to get them dry. 100 microliters of tea buffer were added in order to resuspend the samples. Finally, both tube samples were placed into the fridge for 48 hours.
Discussion. The CTAB protocol was followed along the whole laboratory practice. Even though we previously analyzed each step, the practice took longer than expected. We believe that this occurred due to the lack of experience with the required methodology for the DNA extraction from the cedron tissue sample. Some decisions had to be taken for the preservation of the DNA sample. Because of the unexpected extension of the practice, the samples were left in the freezer for several hours 2 times. The selection of the most suitable step for each of the storages was chosen to protect the DNA from the different molecules present in that specific moment. For example, if the DNases were left with the DNA, it would have been degraded, or if the pH was not stable, it would have affected its integrity. After all the procedure, the pellets from each of the samples look completely clear. This result could mean 2 things. The first one is that our DNA is pure, and the second one is that we lost DNA during the procedure because of human error, and the pellet is empty. Finally, we expect to observe a clear band after running the electrophoresis gel in the next session.
In conclusion, the DNA plant extraction is a basic protocol in genetic engineering, as it is used as a base for many other processes in genetic manipulation. In this practice, the team learned the essential protocol, which is the CTAB, which fits most plant species. As mentioned before, genetic manipulation in plants is extremely important to solve problems in a 7 billion people world, where hunger, diseases, and the extinction of species are an everyday basis. The genetic engineering of plants and the knowledge of their DNA is basic for making discoveries in pharmaceutics, food industry, biofuels, and many other areas. Whether it is for studying purposes or applications, the DNA plant extraction have helped humanity, where year after year it's getting more difficult to feed, cure, and maintain an overpopulated world. The development of genetically modified plants are to improve the quality of life of humans and many other organisms.